Hi, everybody. Let's get started. I want to thank you all for coming to our Mesulum Center 80 Seminar series today. It's in collaboration with the Department of Radiology. So it's a real pleasure for me to introduce our seminar speaker today, Dr. Eran Dayan. He's an associate professor in the Department of Radiology at the University of North Carolina, Chapel Hill. He also has faculty positions in the Biomedical Research Imaging Center and the neuroscience curriculum there. So Dr. Dayan got his PhD in computational neuroscience at the Weizmann Institute of Science. And then for his postdoc, he moved to the US at the National Institute of Neurological Disorders and Stroke. And then he rose to the ranks of senior research fellow there. At NINDS, he became interested in neuroinformatics and in the use of computational modeling approaches in clinical neuroscience research. He then joined UNC Chapel Hill in 2016, and there he established the Laboratory for Neuroinformatics. So his research uses neuroimaging and a range of modeling approaches to capture mechanisms of resilience and vulnerability in aging and dementia, and he also develops diagnostic and prognostic methods in these populations. Since joining UNC, he's won several awards, including the IBM Faculty Development Award and the Distinguished Investigator Award from the Academy of Radiology and Biomedical Imaging Research. So without further ado, I give you Dr. Dayan. The title of his presentation is Resilience and Vulnerability in Brain Aging. Eran, take it away.
Thank you very much for the introduction, and it's really great being here. So let's begin. 2024 has been a very exciting year in Alzheimer's therapeutic development, including developments in disease-modifying interventions and symptomatic interventions. And this is, of course, all underscored by FDA's approval of donanemab and lecanemab, meant to slow down cognitive decline in Alzheimer's. Now, as we are moving towards an era where healthcare systems, insurers, and individual physicians are going to need to decide who is going to be receiving these disease-modifying therapies, and this is especially relevant if we consider the risk-benefit profile of these diseases. It's very important to start thinking about what is going to be the time point in which we want to deliver these therapies. So here you can see the current phase 3 inclusion criteria for lecanemab and donanemab, and they include the diagnosis of mild cognitive impairment or mild Alzheimer's disease, and a couple of additional screening criteria, including positive amyloid beta findings. But if we borrow from the very famous slogan used in stroke, time is brain, and time is definitely brain also in progressive neurodegenerative disorders like Alzheimer's disease. Now, is this really going to be the most optimal time point in which a disease-modifying intervention may work, or can we detect candidates for treatment earlier? Now, if we want to detect candidates for treatment earlier, biomarkers are likely to help, and we can use or start thinking about using, CSF biomarkers, blood biomarkers, or of course, imaging biomarkers. At the moment, especially for amyloid beta tau and neurodegeneration, but hopefully in the future, also for other disease mechanisms that operate in Alzheimer's disease. But the problem is that in Alzheimer's, biomarkers and symptoms are not aligned temporally. biomarkers are known to kind of develop and accumulate way before symptoms appear, and importantly, there's very large degree of variation among individual subjects, among individuals in this relation between the development or the changes that are seen in biomarkers and future symptoms. And this really poses an issue if we want to detect candidates for disease-modifying interventions early. For me, I always found this as a fascinating demonstration for this mismatch between biomarkers and symptoms in Alzheimer is something that we're seeing in post-mortem studies. So cohort studies that follow up that essentially study cognitively intact individuals at certain ages and then perform post-mortem examinations, they find that up to 40%, 35–40% of individuals who showed intact cognition, again, this is as a function of age, of course, but if we're talking about 75, 80-year-olds who show intact cognition, they may have, or they do show under the microscope, they meet criteria for probable AD. And this is something that we're also seeing. I hope you can see the gray curves here. This is for normal subjects with CSF data or with PET data. Around 40%, when we are talking about the ages of 80, around 40% are going to be amyloid positive, despite showing intact cognition. So all of this suggests that the brain may have certain mechanisms that allow it to maintain function when facing neurodegeneration and the other pathological mechanisms that operate in Alzheimer's disease. And these mechanisms may also be related to what we call the reserve capacity of the brain, some sort of accumulating buildup of resources that then help the brain maintain its function when facing disease or also insult states. So in my talk today, I'm going to focus on reserve and resilience in the brain, and more generally try to understand why some individuals show poor and accelerated cognitive aging, while others appear to me more protected from these adverse outcomes. And more specifically, today I will tell you about redundancy in the aging human brain, which operates as a mechanism of reserve. I will then talk about our approaches of assessing resilience in the brain through simulations. And then I'll tell you about our studies where we modeled variation in the progression rates of Alzheimer's disease with AI and deep learning. So let's begin by talking about redundancy in the aging human brain. Redundancy is probably a concept that some of you are familiar with when you're thinking about engineered systems. So many engineered systems are going to have duplication or redundancy built into them deliberately in order to protect the entire system from failure. And this is something you will find in electronic circuits, server architectures, computer hardware, and so on. But redundancy is something that was postulated to also characterize biological and ecological systems, again, protecting the entire system from failure in some of its parts. Now, how can redundancy help the aging brain or brain that faces neurodegeneration? Let's look at this illustration. So if we're considering a brain like this, where brain regions I and J, which I will start calling nodes in a minute, you will understand why. So brain regions I and J communicate with one another through the shortest path available, which is 2 steps. Now, if the brain faces neurodegeneration and some of the connections essentially disappear, then having indirect connections between brain regions I and J allow them to continue communicating despite the brain facing, loss of some of its parts. Now, a more sparse network like the one you're seeing here, when it faces the same neurodegeneration, nodes I and J become disconnected. So this is the idea that excess in connections in the brain, which we call redundancy, may help the brain cope with a structural damage in the form of neurodegeneration. Now, redundancy can exist in the brain across various spatial scales, going all the way from synapses to networks. And today I will tell you about our approach of quantifying redundancy using resting state fMRI data and diffusion MRI data. Resting state fMRI kind of gives us the ability to talk about what we call functional connectivity, which is just the statistical dependency in the fMRI signal that we're extracting. And diffusion MRI data, we are reconstructing white matter tracts, so essentially real physical connections between brain regions. Now, we are able to quantify redundancy because of what is now a very common approach of representing connectivity in the brain as what we call a graph. A graph in mathematics is a collection of nodes or vertices interconnected by edges, right? So in the past 15 years, this has become a very common approach, maybe a bit less than 15, a very common approach of representing connectivity in the brain. So in graphs, our nodes are going to be brain regions, and I'm going back to also this illustration. This is by all means a graph. So every node here would be a brain region, and the edges, the connections, can either correspond to functional connectivity when I'm using resting state fMRI, or structural connections when we're reconstructing the tracts using a technique called tractography. Now, the graph-based representation allows us to study many topological and organizational properties in the brain, things like the centrality of certain nodes, modularity, efficiency, and so on. It also allows us to quantify redundancy as the, here you go, as the sum of direct and indirect paths between every node pair I and J, going all the way up to L, which is the total path that we can consider, which is kind of dictated by computational, the computational means that we have. So we first wanted to validate this measure of redundancy by asking if redundancy changes as we move along the cognitive aging spectrum. So Steph Langella, a former PhD student, what she did was ask if individuals who are cognitively normal show different levels of redundancy as compared to individuals with early and late MCI. Early and late MCI, this is based on the ADNI data set, which I'm sure the majority of you heard of. ADNI defines early and late MCI based on their performance in this cognitive test. So what we did was analysis of resting state fMRI. The nodes are defined based on a parcellation. What is unique about this parcellation, if you happen to come from the neuroimaging community, is that it combines cortical and subcortical regions. Most parcellations don't. And this also allowed us to focus on, specifically on the posterior hippocampus, which is where we expected to find differences in redundancy between these groups. So how does the data look like? So first of all, when comparing individuals with MCI early and late to cognitively normal controls, we find that individuals with MCI show reduced redundancy in the posterior hippocampus. But you can see that the two MCI groups do not really differ from one another, which really suggests that as we move down the road, across the spectrum of cognitive aging, there's little redundancy left in the system, and you will see more data which is consistent with this kind of hypothesis. And then we wanted to connect these results to cognition, and I will do that essentially in all the studies that I will show you today. We never stop with the imaging data. So we use the composite scores for memory and executive function. So first of all, you can see that the three groups differ nicely across the composite scores for memory, and memory is also nicely correlated with redundancy, and this was mostly driven by the CN and early MCI groups. For executive function in this data set, there were a little bit less differences between the groups, and redundancy didn't correlate with redundancy in the posterior hippocampus, I should say, did not correlate with the composite score for executive function. But you will see that redundancy, when we're considering other systems does correlate with executive function. But at least for posterior hippocampus, we find associations mostly with memory. Next, another study that Steph Langella led. We wanted to see if redundancy was able to compensate, if you will, for structural damage in the hippocampus. So the question was if the very well-known associations that you find between memory performance and volume of the hippocampus, is this going to be mediated by or mitigated by the amount of redundancy in the system? So first of all, we worked with a data set of 102 subjects. First of all, you can see the pairwise associations. So hippocampal volume, of course, correlated very nicely with memory scores. Hippocampal volume also correlated with redundancy, and hippocampal redundancy also correlated with memory. And then we fitted the entire data to a mediation model, and indeed we found that the association between memory and hippocampal volume was indirectly and significantly mediated by the amount of hippocampal redundancy. But what about more global patterns of damage that occur, or not changes, I should say, not damage, that occur as we age? Does redundancy mitigate the impact of these damages as well? So we next worked on a measure called network controllability, which is an interesting measure that comes from control theory and was adapted to the use of brain networks in pioneering work by Danny Bassett's group from UPenn. Controllability asks whether nodes in the brain have the ability to move the brain from target initial states to target states. And this is something which is hypothesized to be crucial for cognitive control, for emotion regulation, for motor control, and so on. And it's also common to differentiate between what is called modal controllability, which asks whether nodes in the brain have the ability to move the brain to hard-to-reach states, versus average controllability, which talks about the ability of nodes to impact easy-to-reach states. And now, what's mostly important in our, in the context of what I'm talking about, studies have shown that controllability is impacted by aging quite strongly. So it was also hypothesized that cognitive decline essentially represents a breakdown of network control. So in our next study, a former PhD student called William Stanford asks if redundancy can mitigate the effects of controllability on the aging human brain. So this was based, controllability is typically measured in structural data and using tractography, so DTI and tractography. So William asked if controllability changes with aging, and then how it relates to redundancy and to cognitive performance. So we first looked at the composition of nodes that had high levels of controllability, and we found that they were distributed across the brain in large-scale networks, which I'm sure many of you have heard of, like the default mode network, the frontoparietal control network, and so on. And then we asked if controllability was also associated with age across single networks, and you can see that in many networks it was indeed negatively associated with age. And we asked if redundancy mitigated associations between controllability and age, and indeed found that in 3 networks at least it did. So redundancy mitigated the impacts that age had on controllability. And then we wanted to tie it all to cognition. We focused on processing speed for 2 reasons. One is the networks that we found to be kind of impacted by age, and the second one was previous work on controllability. So processing speed in our sample, this is, by the way, the HCP aging data set. I don't believe I mentioned it. You can see a pretty significant reduction in processing speed as a function of age. And then we asked if controllability, redundancy, controllability, redundancy, and their combination could predict the extent of processing speed issues that the subjects had. And you can see that the combination of controllability and redundancy, and if we added gray matter volume, it even went slightly above, but not a lot. So it was mostly the combination of controllability and redundancy that were able to predict the extent of processing speed, performance that our subjects displayed. And one final question for this part of the talk. We saw that redundancy is something that changes as we move to normal cognition to MCI. So the question we wanted to kind of explore was, is this really something that just kind of emerges in the system as the brain starts, experiencing cognitive decline or, age-associated volume loss? Or is this going to be something that essentially accumulates across the lifespan? Well, it's been hypothesized to characterize what we call reserve. So reserve is really the accumulation of, or at least brain reserve, is the accumulation of neural resources across the lifespan that helps the brain maintain its function when the brain needs to do that. And this is the hypothesized, a paper by Roberto Cabeza. This is the hypothesized curve that a reserve is supposed to kind of display. So please try to remember it. So what we did, and this was a study led by Mohammed Sadiq, a previous postdoc fellow. We took a data set that acquired data across the entire lifespan. It's called the Nathan Klein Institute Rockland sample. And we took 574 subjects, ages 15 to 85. Reconstructed, I will show you data on whole brain redundancy, but the paper also has single network redundancy data, and it's very similar to what I'm showing you today. We first wanted to see what was the shape of the relationship between redundancy and age. This is what you can see here. And I hope you can see that, I mean, it's not perfect. It doesn't perfectly resemble what Cabeza has shown, but it's pretty close. So redundancy builds up slowly as we get older, and then around the age of 65 starts declining. We wanted to see if other network properties show similar curves. I'll show you 2 network properties here. The paper has probably around 3 more. The network properties I will show you, one is called degree, and this is the most basic graph property or network property that exists. It's just the number of edges that a node has. A second property we studied here is called the clustering coefficient. And this is really just a measure of how much neighbors of a node tend to cluster with one another. You can see tend to form these triangles. So these are 2 very basic measures in network analysis and graph analysis. Both of them showed a much weaker association with age, and they didn't really have that up and down curve shape that we've seen for redundancy. And then again, we wanted to tie it all to cognition, because this is a cohort study and they have dozens and dozens of tests. We wanted to minimize the amount of comparisons and statistical tests that we did. So what our approach was to take a battery of cognitive tests and identify the function that showed the most kind of changes as a function of age, and compare it with a function that still changed with age, but a lot less. So this allowed us to minimize the comparisons of the statistical tests to 2. The one function that showed the most age-associated changes was the color word task, which tests inhibition. The comparison task was verbal fluency. And you can see that redundancy mitigated the impact of age on subjects' performance in the color word inhibition task, such that subjects who had higher redundancy had less negative impact of age on their performance in this test. And this didn't happen in the verbal fluency task. The mediation effect was not significant. So let me summarize the first part of my talk. We saw that redundancy accrues across the lifespan until the mid-60s, mediating the association between age and cognition. We also saw that redundancy kind of depletes from the system or is reduced in the system as we move from normal cognition to mild cognitive impairment. And finally, we saw that redundancy can mitigate the impact of localized and more global changes that occur in the brain as we age. All right, so this concludes the first part of my talk. And let's move to the second part, which talks about assessing resilience in the brain through simulations. So as we move closer to where the brain actually faces injury or disease, we can start talking about what is referred to as resilience, which is it's kind of a vague concept, but you can think about it as the mechanisms that the brain employs in order to maintain function when it faces disease or insult, which of course relates a lot to redundancy, but here we're talking about the actual capacity of the brain to maintain its function. Definitely something that is highly related to redundancy as well. Now, how can we assess this kind of elusive construct? We can't really study it experimentally. Maybe we can with things like transcranial magnetic stimulation, but it's very, very hard to actually have a human subject experience, any kind of challenges such as those that the system undergoes as we age or as we face disease. So one solution for this, to study resilience in an ethical manner, could be through simulations. Now, I want to introduce you to the concepts of error and attack tolerance. This is something that was developed in pioneering work by Albert and Barabasi. And the work was really about complex systems. This was not the brain yet. And Albert and Barabasi showed that complex systems have a pretty incredible capacity to handle what is referred to as error or failure. And that is what's going to happen to the system if nodes stop functioning randomly. But on the other hand, complex systems are very susceptible to what is called targeted attack. And this will be an attack on high-degree nodes. This is all related to one of the most fundamental properties of complex systems, which is their degree distribution. So I'm sure many of you have heard about the 6th degree of separation, experiment, or about small world networks and so on. This is exactly what we're seeing here. So the idea is that complex systems, the brain included, the vast majority of nodes have a very small amount of connections, and a few hub nodes have very large degree of, have very strong connectivity or many connections. And because of that, when nodes stop functioning randomly, they are most likely to hit this part of the long tail distribution and have very little impact on the network itself. However, if we have prior knowledge about the degree distribution of the network and we attack high-degree hub nodes, you see that the network deteriorates very, very quickly. So I actually have another demonstration to show you why this is happening. So imagine that this is a map that represents flights in between networks. And there happen to be 2 storms happening in the US at the same day, on the same day. If these storms hit 2 regional airports, nothing much is going to happen to the passenger traffic, right? In this map, it would be 4.6% of the flights will be canceled. But let's imagine that these, and this is what's going to happen, by the way, in random error or failure, right? The nodes that stop functioning are more likely to be these regional airports. However, this is what's going to happen if the storms hit major airports, and you will recognize the second airport. This will be 60.4% of the flights will be canceled because of these 2 storms. So when we have prior knowledge about the degree distribution, about the connectivity of our system, targeted attacks can be a pretty useful way to see how much the network or the system is resilient. And this is exactly what we did here. And let me shift just for 2 slides to Parkinson's disease. But we studied cognitive decline in Parkinson's disease. This was a study led by a former PhD student, Ariana Cascon. We asked if individuals with Parkinson's disease who experience cognitive decline across time, this was up to 5 years, have different resilience properties using targeted attacks than individuals who do not experience cognitive decline. And as it happens, our 2 groups were pretty similar across things like, if you're familiar with Parkinson's, UPDRS Part 3, which is the motor examination part that is done in the UPDRS scale, and also the Hone-NER stage, which is a staging system used in Parkinson's. We asked whether, we hypothesized really, that a group of subjects that showed cognitive decline will have less targeted attack tolerance than the group who showed stable cognition across these 5 years. So Ariana analyzed resting state fMRI data and quantified a measure of resilience. I hope you can see the graph is a little bit small. But this is done by, essentially what you're doing is you're removing nodes iteratively from the node that has the highest degree to the nodes that have the lowest degree. And the area under the curve, the area under the curve here is really our measure of resilience per individual subject. And then we did, of course, group comparisons statistically. So this is how the data looks like when we look at whole brain attack tolerance. You can see not much interesting things are happening here. But when we're looking at specific networks, and this is specifically the frontoparietal network, there's a really nice difference between the two groups in their attack tolerance. The cognitively stable group had larger attack tolerance than the cognitively impaired group, the group that showed longitudinal changes in cognition. And what was interesting in terms of the frontoparietal, or I would say extended frontoparietal network, this parcellation again looked at also at associations between the cortex and subcortical regions, and the extended frontoparietal network had many of the nodes that you will find, or many of the regions that you will find in the associative frontobasal ganglia loops that are believed to relate to cognition in Parkinson's disease. But what about, so we talked about attack tolerance in a pretty specific brain network that is related to cognition in Parkinson's disease, but it was still a pretty specific set of findings. We next wanted to understand if the overall organizational properties of the brain also contribute to resilience and to resilience against attack simulations specifically. And we did this through a technique called K-shell decomposition. It's a little bit complicated, but what K-shell decomposition, and I will return to this, so we don't have to understand it in this slide yet. K-shell decomposition reconstructs connectivity in a network into a set of shells of increasing connectivity. Now, why did we use this technique? Because of a very interesting paper that was published that showed that resilient systems have two properties. One is, and think about a big city right now and kind of economical structure of businesses and companies that operate in a big city. I think this could be a nice way to understand what this paper is showing. They showed that resilient financial systems have two properties. One, it's a very strong core. The core is the shell that has the highest degree of connectivity, okay? So the core here is composed of multiple financial sectors that are interconnected with one another. But what this paper found is that it's not just the core, also the outer shells, the layers of the system that have the lowest degree of connectivity, they are also very diverse, and they also have a nice connectivity between them. So think about financial sectors in cities like Chicago and New York and so on. You will probably have, a few industries that form the core of the financial system in the city and kind of talk to one another and collaborate with one another. But to have a resilient financial system in a city, you also need the more peripheral parts of the system to talk to one another and be diverse enough in order for the system to maintain its resilience. And what they did, this is a really nice demonstration, they try to understand what's happening to the system when this is lost. And I hope you can see this is, the font here is pretty small, but these are things like Black Monday in 1987 and the collapse of the financial crisis in 2008 and 9, which probably many of you still remember. So it's very interesting. They kind of validated this approach against what's happening at the macro level of the financial system. So William, who is really, I would say, scary smart, wanted to see what's happening in the brain if we use this approach. So the idea was, again, to take data from a large data set, like the Human Connectome Project Aging. This was based on resting state fMRI. Again, previous study by William was DTI-based. So what William did was, we reconstructed brain connectivity, did causal decomposition, which you will see again in a minute, and then assessed how it's related to cognition and also to specifically how aging impacts cognition. So causal decomposition, again, the idea is that we are decomposing the connectivity in our system into shells of increasing connectivity, such that what we call the one shell, the outer shell, has a degree of 1. Second shell has degree of 2. Just reminding you, degree is the number of edges that every node has. And then you go all the way up. Here it's 3, but you'll see that in our brains it's a lot more than 3, to the core of the system, which is the shell that has the highest degree. And then what William did was he combined this with attack simulations, removing nodes according to their degree, and kept track, this is like that paper that we saw before, kept track of the value of, K is the degree, right? The K value of the core of the system, and this gave us a measure that of the resilience of the core of the entire system, the max core resilience measure. So what we found as very interesting was that the brains actually had an organizational kind of structure that resembled those financial systems that we saw before. In other words, very diverse cores and also diverse outer shells. And the second thing that was interesting was that the Kmax resilience measure correlated exactly with everything we expected it to correlate against. So first of all, with age, then with memory, and then again, we used the mediation model to see if the associations between age and episodic memory were mitigated by the amount of max care, were mediated, sorry, by the amount of max core resilience, and indeed they were. So I'll summarize the second part of my talk. It was a little bit shorter. The third part is longer. We saw that attack tolerance in the frontoparietal network is predictive of protection against cognitive decline. And then we saw that brains display a core topology organization that was shown to promote resilience in other complex systems, and that core resilience specifically is also associated with better cognitive function outcomes in aging. All right, how am I in time? I think I have enough. Let's move to the third part of my talk, which kind of brings us closer to dementia. And here we focused on variation rates as we move from mild cognitive impairment to dementia. We wanted to kind of cover, I wanted my talk to cover the entire spectrum in a sense of cognitive aging. So now we're moving closer to dementia. And we used specifically deep learning approaches to try to understand something new, I hope, about progression rates from MCI to Alzheimer's, also progression rates within Alzheimer's, which I will show you a little bit later. So this part of my talk focuses on MCI, which is a very heterogeneous construct. We know that certain individuals with MCI progress to AD, some progress to non-Alzheimer's dementia, some progress or end up showing neuropsychiatric dysfunction and not even dementia. And also within people who have MCI and do have gradual cognitive decline, progression rates are very, very different. So there's been attempts in the literature to constrain this heterogeneity. And up to now, they were mostly based on cognitive or neuropsychological subtyping. And this is the idea of subtyping people based on their performance in neuropsychological tests and tasks. Now, these are very successful approaches, but the one thing that they do not do is really consider, or necessarily do, is consider kind of the biological definitions for Alzheimer's and MCI, which we are now attempting to use more and more, such as those proposed by the ATN framework, if you're familiar with it. But the ATN framework doesn't do a very good job in subtyping MCI or constraining the variability shown in MCI. And this is especially true, and if you're not familiar, I'm sorry, ATN framework means it's a framework that uses biomarkers for amyloid beta, tau, and neurodegeneration. You can see that neurodegeneration, the N is in parentheses, and the reason is that it's not really specific to any of any of the stages that we're seeing here. But we actually wanted to specifically focus on neurodegeneration and ask if we can use the powerful capabilities that AI and deep learning give us to use neurodegeneration and be able to constrain MCI. So an approach that a former postdoc of mine, Ki-Chang Kwak, developed does something like this. So essentially, it's a deep learning model, a convolutional neural network, that we first train to differentiate between amyloid and tau positive Alzheimer's, individuals with Alzheimer's, and amyloid and tau negative controls. So the model learns to differentiate between these two groups. Then what we do is we kind of trick the model, if you will, and we give it MCI, data from MCI. This is all structural MRI, by the way, biomarker for neurodegeneration, for atrophy. So we trick the model, we give it MCI data, and then because it was trained with two labels, it has to decide, I think that I'm seeing AD, or I think that I'm seeing MCI, right? And then we call it MCI-AD and MCI-CN. So MCI that has atrophy patterns which are more AD-like, or atrophy patterns which are more CN-like. And then what we did was we validated these labels. So essentially, first of all, we gave the model 380 MRI images of MCI, of individuals with MCI, and it decided that 67% were MCI-CN and 33% were MCI-AD. We validated these labels against multiple biomarkers, including CSF biomarkers for amyloid beta and tau, FDG PET, which is used as a secondary measure of neurodegeneration, amyloid beta PET, and also against things like CDR sum of boxes and ADAS scores, which are used for staging and kind of global assessment of cognition in Alzheimer's. So everything we gave the model, every biomarker that we tested these labels against showed very nice differences between the MCI-CN and MCI-AD groups. But then, critically, I would say, we always care about what's going to happen later, right, about prognostics. So we first tested longitudinal differences in ADAS scores and CDR sum of boxes, and you can see that the two groups show very significant differences as we consider data across 2 years. And probably most critically, we also tested disease-free survival, and the two groups showed very different progression rates to AED, and this was actually also validated in a secondary data set. So, like many deep learning AI models, it's kind of black boxish, right? I'm telling you about atrophy patterns, but we are not seeing anything, right? We don't know what the model is really learning from. So our approach to add explainability to our deep learning models is called occlusion analysis. There's other explainability methods out there. We kind of prefer this one, and I will use it multiple times. Today, you will see. So occlusion analysis essentially occludes part of the image, and then we can compare the performance of the model, of the intact model that learns from the entire brain, to what's happening when one part of the brain is occluded. So we occluded here major kind of systems and parts in the brain, and we found that when we occlude the medial temporal lobe and the lateral temporal lobe, there's very big differences in the performance of the model, and this is relative to when we occlude things like cingulate cortex or the occipital lobe or the frontal lobe, when the performance of the model doesn't really change as much. So, not surprising, right? The MTL is essentially what drove the performance of our subtyping system. In a second study, Kichang wanted to ask if patterns of atrophy in the MTL are also going to be useful for us if we want to predict conversion from MCI to AD. This is a very let me just minimize this. You can see, right? Yeah. This is a very common task in the machine learning AI community that works on diagnostic models or prognostic models for Alzheimer's. We call it the progressive versus stable MCI task, right? So we are essentially binarizing individuals with MCI relative to future data as a function of whether they convert to Alzheimer's or do not. So progressive MCI would be people who progressed to AD, and stable MCI would be people who did not. So again, we used a deep learning model that trains on whole brain structural MRI. We assessed how well the model was able to differentiate between PMCI and SMCI, progressive and stable MCI, and then did occlusion analysis, occluding many, many individual brain regions based on the free surfer program that I'm sure many of you probably heard of, and compared the performance of the model when each individual brain region is occluded relative to the performance of the intact model. This is how the model did with the whole brain. Area under the curve and accuracy was around 74%. A pretty decent performance for this PMCI versus SMCI task. And this is how the occlusion results look like. Delta accuracy would mean that when we occlude these specific regions, the performance suffers more, right? So this is what you're seeing here. And again, we found medial temporal lobe and hippocampus and related regions to be mostly the ones that contributed the most to the performance of our prognostic model. But what about, and we saw that the hippocampus appeared in that model, but what about more specific parts in the hippocampus? The hippocampus is a pretty complex structure composed of multiple subfields and substructures. And there's inconsistency in the literature regarding which of the subfields is implicated in MCI and AD. This is just a table that summarizes 21 studies. I should probably update it. It's my own table. I should update this, but you can see a pretty big inconsistency, among individual papers. So another study by Kichang, we used the exact same approach that I showed you now, but this time including individual subfields in the hippocampus, which you can see here. And the results revealed actually very large differences between the subfields. So when we occlude things like the CA1, subiculum, and presubiculum molecular layer, there's pretty big differences in the performance on the model. But then again, when we occlude other subfields, including very big ones, the model doesn't really suffer much. Performance doesn't change that much. This, if you think about it, may relate to redundancy as well, meaning that some parts of the hippocampus are not really related or do not really